Globodera rostochiensis, it develops cysts on potato. Seed gall nematode is Anguina tritici. Spiral nematode is Helicotylenchus species. The most commonly occurring root knot nematode in India is Meloidogyne incognita. The nematode associated with white tip disease in rice is Ephelinquates baceae. Valval cone is present in Anguina species. Plant parasitic nematodes generally attack roots. Virus transmitting nematodes are Ziphinema and Longidorus. Ufra disease of rice is prevalent in northern India. Citrus nematode causes slow decline of citrus. Plant parasitic nematodes possess three-part esophagus, valved basal bulb. Pratylenchus coffee is an important nematode pest of coffee. Nematode body is bilaterally symmetrical. First separate division of nematology in India was established at Punjab Agricultural University, Ludhiana. First report of a plant parasitic nematode in India was in the year 1901. Ditylenchus species forms nematode wool. Pratylenchus species does not produce eggs in gelatinous matrix. Heterodaptitis indicus is an example of entomophilic nematode. The length of most plant parasitic nematodes ranges between 0.1 to 1 mm. Giant cells are produced by Meloidogyne species. In root knot and cyst forming nematodes, the effective stage is second stage juvenile. The most important general symptoms of plants affected by nematode is stunting and yellowing. Malloya disease of wheat is caused by Heterodera aveni. Slow decline of citrus is caused by Tylenculus semipenetrans. Source of inoculum of earcockle nematode disease is seeds. Simulation of mitosis increasing numbers of cells is called as hyperplasia. The most susceptible group of crops to root knot nematode is vegetables. The body cavity of nematode is called as pseudocoelom. The nematicide which can be applied with irrigation water is nemagon. Number of life stages in nematodes are 6. Listed nematodes are migratory endoparasites. Burrowing nematodes in India are serious pest of banana. The feeding areas in plant roots induced by root knot nematode are known as giant cells. The root knot nematode wilt complex in chickpea is Meloidogyne incognita or Meloidogyne javanica with Fusarium oxysporum form species Caesari. The cereal cyst nematode is Heterodera aveni. In brinjal and tomato, the wilt disease complex consists of Meloidogyne incognita and Fusarium oxysporum form species Lycopersici. One of the examples of seed-borne nematode disease of rice is white tip nematode, that is Ephelinquates baceae. The causal organism for Ufra disease of rice is the nematode Ditylenchus angustus. Yellow carrot or tundu disease of wheat is caused by Clavibacter or Corynebacterium and Anguina triticum complex. Kalahasti malady is caused by Tylencorhynchus brevilineatus in groundnut. Organic soil amendments are able to reduce the nematode damage by stimulating the microorganisms antagonistic to plant parasite nematodes. The mushroom nematode which cause problems in mushroom production is Ephelinquates agaricus. Wheat gall nematode is Anguina triticum. Citrus nematode is Tylenculus semipenetrans. The vector nematode of virus disease in grapes is Xiphinema americanum. Malloy disease is caused by Heterodera aveni. Grape fan leaf virus is transmitted by Xiphinema index. An important nematode pest of mushroom is Ditylenchus myceliophagus. The stage of the nematode which cause infection is J2 stage. Infective stage of entomopathogenic nematode is again J2 stage. 4 larval moltings are observed in nematode life cycles. In cotton, nematode entry associated with the fungi of Fusarium species. Entomopathogenic nematodes contain bacteria and these belong to the genus both Xenorhabdus and Photorhabdus. NETU, native, is transmitted by the nematode genus Rhotylenculus reniformis. Odontostyle with 3 large knobs is present in the nematode Ephelinchus. Important nematode which attacks vegetable is Meloidogyne species. False root nematode is Nacobus species. Marco is found on the tail tip of nematodes belong to Hersmaniella species. In male nematodes, the common opening of both testis and intestine is known as anus. Citrus nematode was first discovered by Siddiqui in the year 1961 in India. The potato cyst nematode in India was reported first in the year 1961 by F. W. G. Jones. Carter in the year 1943 discovered nematicidal properties of DD mixture. The circulatory system is absent in nematodes. The excretory system of Caesarensian nematode is tubular. The suppression of root knot nematode in the rhizosphere of some species of Tagetes is attributed to the release of alpha terthionyls. The quiescence of nematodes under high levels of salinity is osmobiosis. Majority of the economically important plant parasitic nematode belong to the group Tylenchids. A crop variety that suffers no damage but allow high rate of nematode reproduction is regarded as tolerant. The retrose annulation is seen in some genera of criconematids. In fixatives for nematodes, acetic acid is added to alcohol because it counteracts the shrinkage caused by alcohol. Juvenile stage of nematode do not feed on plant roots, that is Rhotylenculus reniformis. Red ring disease of coconut is caused by the nematode Barsaphilencus cocophilus. In Behrman funnel technique, the nematodes are extracted with the help of their body shape and movement. Nematode which feeds on fungus are Ephelinquates humatum and Ephelincoids compositicola. Nematode having the narrowest host range is Heterodera cajuni. Nematode taxonomy has been mainly based on morphology. The gelatinous egg matrix of nematodes is helpful in survival in drying soil. Nematode can survive for many years without water in the state of anhydrobiosis. Pasturia penetrans is an obligate parasite of nematode. Farmers are mostly ignorant about the damage caused by the nematodes in their field because the nematodes are not visible to the naked eye. Nepoviruses are transmitted by dagger nematode. Only quarantine nematode pest reported in India is Globodera rostrociensis. Biofumigation of soil is a technique in which green mustard plants are burned in soil under cover. Nematode head is radially symmetrical. The only tylinkid nematode having rectal gland is Meloidogyne species. Tundu disease of wheat is caused by Anguina tritici with association of the bacteria Clavibacter. The most preferred crop for cereal cyst nematode is barley. In root knot nematode attack, giant cell is formed by both hypertrophy and hyperplasia. The cysts of Heterodera aveni remain viable up to 5 years. In India, root knot nematode was first reported by Barber in the year of 1901. Asymmetrical type of excretory system is present in the members of the order Tylinkida. Nematode which causes pearly root disease of PGNP is Heterodera cajuni. Seed nematode is Hemicyclophora. Nematode which causes slow decline of citrus is Tylinculus semipenetrans. The strain used for staining nematodes within the roots is acid fuscin. Major nitrogenous excretory end product in nematode is ammonia. Specific gravity of nematode is more than specific gravity of water. Polymethyl transaminase, that is PMTE, has been reported in Ditylincus dipsaki. Morphometry is a system of measurements introduced by D-Man. Root knot nematode in tea from South India was first reported by Barber. Scientific name of root knot nematode in tea is Meloidogyne brevicorda. Neural tooth is characteristic feature of Monuncus alternatus. Fratylincus dilateri is not a quarantine nematode pest. Infective stage of reniform nematode is young female. Trichodorus is not a migratory endoparasitic nematode. Anguina millefoli, this nematode produce leaf galls. All nematode is AWL, all nematode is Dolichodora species. Anhydrobiosis most commonly and for longest period is reported in Anguina tritici. The three regions of nematode spicules are capitulum, corpus, and lamina. Excretory pore is located slightly anterior to vulva in the genus Tylinculus. Bursa in males is absent in Trichodora species.